where I am in Boston. I live in Sharon. That's behind me, Sharon, Massachusetts.
Great. Well, I'm delighted to welcome everyone today to our special research presentation from Dr. Steve Schachter. I met Steve a while ago through our common research interest in epilepsy and in kind of homeostatic therapies for epilepsy, and recently corresponded with him more during my Charles Bullard Fellowship at Harvard Forest. So Steve did his undergraduate work at Case Western in Chicago, and he was originally a chemistry, biology, and psychology major, but in finishing early, he could only finish his chemistry degree, but he definitely has cross-disciplinary interests. He's currently a professor of neurology at Harvard Medical School, and he's done a lot of work. He's spent a lot of his career with patients with epilepsy and also research on botanical approaches. He's probably not going to spend a lot of his time on it today, but if he wants to share anything as well about his role in leading a big NIH study called REDEX to develop treatments for COVID-19, that's another exciting new thing that he's doing in his career. So without further ado, I hope that you'll help me give a warm welcome to Dr. Steve Schachter.
Thank you. Thank you. That was quite warm, as warm as it can be on Zoom. Thank you. Okay, well, Susan, I really appreciate the invitation to share some time with you and your students and colleagues today. It's a real pleasure for me to do that. Plus, it brings back a lot of nice memories about when I, as a medical college student, had lectures from people that I thought were pretty old. So it goes full circle. That's the lesson I'm learning today. So at Susan's invitation, I put together a talk based on my own personal experience relating to potential neurological applications for botanicals, and specifically some botanicals that grow naturally in forests. I got into this at Harvard Medical School, which might seem like a contradiction in topic versus culture. A number of years ago, the medical school, thanks to a large gift, established what you might call an alternative therapies division. It had a much longer name than that. And that was brand new for Harvard Medical School. And part of that effort was the study of natural products, particularly botanicals. But it also embraced a variety of other therapies that are typically not taught or were not being taught in medical schools at that time, but which have been widely adopted, some of which are shown here. And I got involved with this group at the invitation of the director at that time because I was intrigued by the connections that he had with botanical experts, particularly in Hong Kong and Japan. And it opened a whole new world up to me. At that time, I had been treating people with epilepsy for, let's say, about 25, 30 years. I conducted a number of clinical studies into new therapies that had been synthesized in laboratories that were not natural in their origin. And despite all the progress or all the work put into developing new therapies for epilepsy, still about 2 out of 3 people with epilepsy either have significant side effects or simply have no benefit in terms of seizure reduction from existing therapies. So I wondered to myself, well, maybe there's some potential for botanicals with regard to therapies for epilepsy. And that was not a new thought at all. I mean, prior to the advent of bromides, which is a chemical about, let's say, 100 years ago, doctors were using botanicals all the time to treat epilepsy, and there's some very well documented literature in the Western medical tradition, and well documented literature in traditional Chinese medicine and Ayurvedic medicine going back centuries. And the advantage for somebody who's interested in botanicals for epilepsy, the advantage of reading these old texts is that a seizure, and many of you may probably have seen somebody having a seizure, a convulsive type of seizure. It's unmistakable in the way it looks, and it looks the same today as it would have looked, you know, when the Yellow Emperor's textbook was written centuries ago. So if something was taken by mouth or by some other formulation and it stopped those convulsions, it would have been obvious to somebody back then. It's not a subtle, you know, diagnosis made based on an X-ray or something that was unavailable back then. And a number of notable people have been treated for their epilepsy with the botanicals. And I'm showing here the cover of a book. And on the cover of the book is a painting that was done by Van Gogh. It's a friend of his named Gaucher, who was a physician. And this portrait hangs in a museum in Paris, not the Louvre, but the other one. And you see a plant on the table. That's the foxglove plant. And foxglove plant grows along the edge of forest. You can also plant it and cultivate it.
It's in my ears.
I'm sorry?
Integrative therapies, natural products.
Okay. Susan, I don't know, you're muted, but maybe that's on purpose. I don't know. So anywho, the foxglove plant is a source of a compound called digitalis, which works on the heart, but it also is an anti-seizure medication. And Van Gogh himself was treated with digitalis. So there's a rich tradition of the use of botanicals, which you might also call time-honored, in treating epilepsy. So I want to quickly review why it is that we need new therapies for epilepsy, whether they're botanical or not, just to show you the urgency of the need, and if we find something from a botanical, the potential benefit it will have to millions of people. So about 60 million people in the world have epilepsy. Many of them also have other issues, whether it's depression, anxiety, or cognitive difficulties. These all are also related to the disordered brain function from which the seizures arise. As I mentioned a moment ago, the majority of people with epilepsy do not have satisfactory treatment with now more than 20 available drugs on the market and devices. And consequently, they're at risk of having seizures, which can range in consequence from being socially embarrassing to being fatal. And they're at risk for the consequences of side effects as well, whether that's double vision, loss of balance, trouble with memory, and so forth. So there's a huge unmet need among people with epilepsy for an effective, safe therapy. Furthermore, the estimated 40 million people in the world with epilepsy who live in places like West China, Sub-Saharan Africa, rural India, access to pharmaceuticals is very inadequate. And on top of that, there's a cultural bias against Western drugs in many of these areas. And thirdly, even if they had access to it, they can be unaffordable. A potential benefit of botanical therapies is that they would be culturally acceptable and potentially much more affordable when scaled up compared to pharmaceuticals. So I got very interested in this, given the unmet need and the opportunity. And as I came to find out, that there was already a very long history of looking at botanicals as medical therapies, including neurological therapies. I mean, some of the, over half of the best, or over half of the most prescribed drugs in this country have botanical sources. An example of a drug in the neurological space that's a plant is shown here. This is a plant called the common snowdrop, from which galantamine is isolated. And galantamine is an approved therapy for Alzheimer's disease. It's available over the counter as well. You could buy it as a dietary supplement. It works on the same system in the brain as does another drug that I will be discussing later, acetylcholinesterase inhibitor. And it's thought that this plant was what Homer was referring to in his book, The Odyssey, that sailors would eat in order to remain cognitively sharp. So as I got into this new area to me, I came to realize and to understand that quite a few botanical herbs around the world, many growing in forests, have been used historically to treat epilepsy, to treat the seizures of epilepsy. And laboratory scientists had studied many of these to look for what it does to brain cells that could support its clinical use. In other words, does it work on excitatory or inhibitory neuronal function? Does it have anti-inflammatory actions? Does it change pH? What is the biophysical effect of these herbs on the brain? And most of that work, of course, is done in animal studies or on cultures of neurons. So the very first thing we did was to conduct a large literature search to try to find out which specific herbs had been tested clinically or in the laboratory and had a tradition of use in epilepsy. We came across a number of different studies, not surprisingly, because many of these were not carried out in major medical centers. The study designs were not sufficient to make any definite conclusions about safety and efficacy for any herb in the field of epilepsy. And by and large, that's true across the therapeutic spectrum, not just in epilepsy or neurological diseases. But these studies can be very difficult to do, particularly when you're using extracts of herbs and it's hard to control for the constituents of those herbs as opposed to a single compound. And it's also difficult when the therapy is administered in the study as part of a more holistic medical approach, meaning that more than just the therapy can be varied from one person to another in the study. So that makes it almost impossible to assign cause and effect in a prospective study. But this literature review is very good for us to come up with the most commonly used and cited and referenced and studied herbs. And this is in the South Asian literature. And that gave us a starting point. And so what we did, and what further excited us, that just approved a new approval pathway by which botanical products could be approved by the FDA for therapy by prescription. What you see on the right here in the picture is the constituents of a therapy for stroke that's been used in China. And if you show that to a physician like myself, 15, 20 years ago and said, would you write a prescription for this? I always said, no, because I don't know what I'm giving the patient. I don't know how to measure it in their body. I wouldn't know how it would interact with other drugs they're taking and so forth. So what the FDA did was to create a very robust process by which an extract, which they called a botanical drug product, could be approved. Now, typically what the FDA approves in a drug is the compound. They approve its manufacturing, its the synthesis, all the various chemical steps involved. That's not possible with an extract. So what they approve is a process by which the raw plant is converted into the extract. And the manufacturer has to put together a process that's sufficiently robust so that the outcome of it, no matter what the growing season, what the year, the outcome is the same. And they can't possibly approve the compounds within those within the extract because there's dozens, if not hundreds or more compounds. For example, this formula for a stroke product has all of these compounds in it. And you couldn't possibly, understand the pharmacokinetics, the pharmacodynamics, the drug interactions, all the kinds of things that most Western doctors worry about when they prescribe a single molecule, a single compound. So it looked like an opportunity where there could be a viable regulatory pathway to commercialize extracts for epilepsy, which is important because bringing a product to market is very expensive. You have to have a company that's willing to put the money into it and commercialize it. And part of that is having the possibility of an FDA approval. And for developing countries, it seemed possible, if not likely, that botanically derived therapies could be culturally accepted, perhaps even grown in local areas where they would be consumed and would be therefore affordable. So with all that, we started something we called the Harvard Epilepsy Botanical Program. And we had several goals with that program. First was to get hold of herbal medicine extracts that there was evidence from laboratory studies or clinical work that they may have some potential use in epilepsy. The second goal was to isolate compounds from those extracts and test them in the same modern methods that we tested the extracts. And then the third and fourth goals were then to move these products into clinical testing and eventually into FDA regulatory pathway with a view towards commercialization and use by people with epilepsy. We put together a global team of experts with complementary expertise, from being experts in botanicals, natural product chemists, neuroscientists, animal experimentalists. Literally it stretched from China to Senegal in Africa, Korea, different parts of the United States. This was an interesting process to put together, and a wonderful team. Here you see where some of these folks were located. And together we had a broad range of experience from growing, from identifying plants, species, subtypes, through to extraction methods, to bench research using patch clamp techniques some of you might be familiar with, to testing extracts in animals, and then even into clinical studies when possible. So we spent a lot of time figuring out, well, how are we going to select these botanicals? I mentioned we did the literature search. We wanted to be testing what we thought we were testing, so there were a variety of authentication methods that we had to use to do that. We had to be careful about the extraction method we used to get extracts out of raw plant. some of you might be familiar with this chemistry, but there are different mechanisms by which you turn a leaf or a root or a stem into an extract. And apropos to Dr. Seraphin's question about pharmacokinetics, this is really important when studying botanicals for neurological disorders, where you want the agent to cross into the brain, across the blood-brain barrier. Because if you use water to extract chemicals from plants, you're going to miss alkaloids. Alkaloid, that's a category of compounds which are highly lipid soluble, meaning they enter into fat cells or cells with fat in them, and the blood-brain barrier accepts lipophilic compounds and moves them into the brain. So it's important for this work that alcohol or methanol-based extraction methods, which preserve alkaloids, are used. So these are some of the details that I had no clue to until I started getting into this work. And then once we had the extracts, then we wanted, you might say, well, why didn't you start testing them in people with epilepsy? And in fact, that's kind of the approach taken in some places around the world. But I was trained, and I support the concept of testing potential therapies in animal models first, for a number of reasons. One is you get an idea of what the dose may need to be in people. And you get an idea of the range of doses or the range of blood concentrations that could be tolerable and potentially effective. And from a very pragmatic point of view, it's very important in terms of securing follow-on funding or any kind of funding or the interest of any kind of commercial entity, because they're accustomed to drugs being first tested in these animal models. So long story short, and this is just going into further detail about what those clinical studies would be. So long story short, we tested just about 40 different herbal extracts in well-established animal models relative to epilepsy. Now, when I say well-established, it was a NIH-sponsored laboratory that did the work out in Utah. Same laboratory with the same methods, screen compounds supplied to them by drug companies as potential therapies for epilepsy. So they had tested tens of thousands of compounds. We were the first to send them extracts, which I found interesting. And we also tested some pure compounds as well that we had derived from these extracts. So what we found was that nearly 3/4, 70% of these materials worked in these animal models of epilepsy. When I say worked, they prevented seizures from occurring in these animals. And the other, which you might wonder, well, why didn't 100% work? And that's a good question. But I would suggest, I've been told by drug companies that send libraries of compounds to the same laboratory that they're lucky if they get 5% hit rate. So we took this as evidence in support of the process of honoring the tradition of use of an herb for epilepsy, and then testing it in a modern validation for activity in epilepsy. But you might wonder again, why didn't they all work? Well, first of all, some of the reports just may have not been accurate. But secondly, these animal models are not the end-all and be-all of validating that a therapy might work for a person who has epilepsy. they have their limitations. And I have a feeling that some of the remaining 30% of the materials would be effective in people with epilepsy and would work in ways and mechanisms in the brain that are totally different than everything else that we've ever brought to the market as therapies. But the catch-22 here is that no one's going to pay for all the work that it takes. No one's going to take a chance on this without it having shown activity in animal models. So, and the other very interesting thing that we saw was that the ratio of the dose in a rodent that caused side effects was much high, that the blood concentration at which side effects occurred was much higher than the concentration at which seizure benefit occurred. And that ratio we call the therapeutic window was much higher with these extracts than is the case with single drugs. And that was very interesting because it's consistent with traditional Chinese medicine principles, which is that the mixture of compounds both treats the disease but also supports the host, meaning that it makes the therapy more tolerable. We then started to look into mechanisms. How are these herbs working? And most commonly, they are antioxidant, which was interesting because oxidation is a trigger for inflammation, and inflammation in the brain has only been recently pursued by neuroscientists as a mechanism underlying the genesis of epilepsy and the continuation of epilepsy. Some of them also inhibited the excitatory neurotransmitter effect in the brain, that of glutamate. And others, as I'll describe in a second, with one called huperzine, worked on the inhibitory neurotransmitter system called GABA. So we tested about 40 of these things. The very first one I sent off was actually called huperzine A. And I want to focus in on that as a case study of a forest botanical for a neurological condition. So those of you who've trained in medicine, you might have heard the expression bench to bedside. That means you discover something in a laboratory and you bring it to patient care. I think the way I would sum up what in this epilepsy botanical program is from tradition to bench to bed. So let's talk about huperzine A. Here's a plant from which it's extracted. It's called Huperzia serrata. In Chinese, it's called Chengseng Ta, which translates to 1,000 pagodas. You can see the leaf structure here. This is a fern moss. The leaf structure, you can imagine somebody thinking that looked like the roof of a pagoda. You know, the many, you've seen pagodas that have many stories to them. This is the molecular formula, or the structure of huperzine A. To chemists, it's interesting. It has 2 chiral centers, which makes it pretty difficult to synthesize. It has a tradition of use in China, particularly for treating fever, swelling, inflammation, and more recently for what may be schizophrenia. It grows at high altitudes in China, in damp forests. It takes 10 to 12 years to mature. It's not amenable to being a crop that could be grown for medical purposes. You remember that picture of the common snowdrop I showed earlier? That actually is grown in good agricultural practice farms in Canada because it lends itself to agricultural, to cultivation, but that's not the case for this plant. Um, it has a very complicated chemical formula. It's a relatively small molecule. And before we got involved with huperzine, it was known to be an inhibitor of a enzyme in the brain called acetylcholinesterase, which is responsible for breaking down the neurotransmitter acetylcholine. And it also, and with regard to that mechanism, it has comparable potency to 4 other compounds that are on the market for Alzheimer's disease, including galantamine, which I showed earlier, because that too is plant-derived. But when I first got involved with huperzine, I discounted its acetylcholinesterase mechanism because none of us in the field were thinking that the cholinergic system was at all related to seizures. We knew it's related to memory and to the immune system, but if anything, one of our models of seizures is to give the cholinergic agonist called pilocarpine, which causes it. So I thought that this was an interesting but unrelated mechanism when it came to epilepsy. What we did find, though, when we sent huperzine, but sorry, the reason I studied it was because it was said in the literature to be an antagonist of glutamate, the main excitatory neurotransmitter in the brain. So that's a mechanism that's relevant to stopping seizures. So that's the reason I studied it. And the subtext here, by the way, is accident favors a prepared mind. And that accidents are no accident, or not really accidents. Now, maybe I can explain why I say that in a moment. But when we studied huperzine in a variety of different well-established animal models for epilepsy, we found it to be effective. In fact, I got a call from the head of the laboratory immediately after he got these results, and then he said, "What is this?" He didn't know the name of it because we anonymize compounds and extracts when we send them for testing. And I told him, and he said, "This is the most potent compound we've ever tested in this laboratory out of the tens of thousands of other compounds." So I thought that was interesting. And its relative potency to other epilepsy therapies is shown here for one particular model, the 6 Hertz model. And the details of this model aren't important, but underscoring its potency, you see that in this level of seizure induction, intense electrical intensity, it's effective at 0.78 milligrams per kilogram in the mouse compared to orders of magnitude more with these other established drugs. So we got very excited about that. We tried to figure out why is it working, and I'll just tell a short story here. It turns out that it doesn't, at these concentrations where it's effective in the seizure model I just showed you, at these concentrations and the projected concentration in the brain, because there had been work on that already, there was no way that it was inhibiting the glutamate receptor, because we knew what the what's called the binding affinity for huperzine is. So we concluded it's not working in that way. Well, when I got the results back from the NIH about how effective it was in stopping seizures, they also noted that the animal was having some tremors, shaking. And the experimenter said, it looks like a cholinergic effect in the periphery, in the body, because there's acetylcholine released in the body as well, not just the brain. And that's broken down by a different enzyme called butyrylcholinesterase. But when you inhibit that enzyme in the animals, they can have some tremors or shaking. So I said, fine, let's co-administer huperzine with a anticholinergic drug that is active only in the body, that doesn't cross into the brain. And then they came back to me with the results. And what they showed was, yes, the shaking did stop. But what they also showed was that the seizure protection was no longer present. And I thought, well, how could that be if we gave huperzine, but we added something that doesn't get into the brain? Well, it turns out that they gave atropine, a compound called atropine, as the other medication. And it turns out atropine does get into the brain. And so because it can cause a psychosis, actually. It can have central nervous system side effects. So was atropine blocking the effect of huperzine in the brain to stop seizures? And I said, how is that possible? Is there really an acetylcholinergic, is there a cholinergic contributor to seizure protection? So we did some digging, and this would have made a great assignment to the neuroscientists in the group here. We did some digging, and it turns out that on a type of brain cell called the GABAergic interneuron, which is responsible for setting the inhibitory tone of the cortex, turns out that on that neuron are cholinergic receptors for acetylcholine. So when acetylcholine is released from one neuron and binds to the acetylcholine receptor on the GABAergic interneuron, that GABAergic interneuron releases GABA at its terminal, which is in the cortex. So that would have an inhibitory effect. If you co-administer atropine at the same time as huperzine A, atropine is going to block that receptor from acetylcholine having that effect. So we then did some work with a technique called transcranial magnetic stimulation, which I won't go into details, but it basically provided very good supportive evidence that huperzine increased the GABAergic tone in the cortex. So we thought, well, this is an interesting and plausible mechanism by which huperzine could stop seizures, taking advantage of the brain's intrinsic inhibitory network, not by directly stimulating the GABA receptor, which is what drugs such as phenobarbital and Valium and other drugs in the benzodiazepine class do, which can have significant side effects of sedation and also risk of addiction or becoming dependent, and also a risk of down-regulating the GABA receptors and therefore requiring higher doses. So now we had a mechanism by which we could think of how huperzine worked in the brain, and that led to some other exploration of other potential neurological uses for huperzine A, including pain. And we then did a series of experiments, again, using animal models of pain to suggest that huperzine could be a very potent way to treat pain, not only neuropathic pain, but pain arising from pathology in the brain, to garden variety, step on a nail type of pain, was called somatoform pain. Then, again, based on this mechanism and particularly how potent it is in blocking acetylcholinesterase, we tested it in an animal model of nerve toxin exposure, which periodically you hear in the news of somebody was poisoned or died from exposure to nerve toxins. This is one of the main chemical threats that governments, militaries worry about. Sure enough, huperzine was more effective and better tolerated than galantamine, which is one of the other drug that's been shown effective in these animal models. And before we ever got involved with huperzine, it was already being studied in Alzheimer's disease and myasthenia gravis, which is not a brain disorder, but it's a disorder of muscle that results from impaired conduction of acetylcholine from motor neurons in the body as they project onto muscle tissue. That's an autoimmune disease. So this one forest plant, Huperzia serrata, now appears to have many potential neurological uses. And that's, I only give this example in detail because it's the one I personally have spent the most time with. And it can't be hugely beginner's luck because we know other plants that grow in forests that have been used traditionally in epilepsy as well. But I would not have picked it. I would not have gotten involved in it had it not been for a reason that was not true. I mean, if it hadn't, so serendipity, like I say, I mean, we set up this evaluation process, you know, in a very definitive, structured way, and yet something came into it called huperzine, which I latched onto some basic science information that turned out to be not at all relevant to why I was interested in it. But we pursued it, and it's now in the hands of a drug company that's being developed for clinical use. So that's the end of my talk. I hope I gave you an interesting story to think about, some lessons learned. I'm more than happy to answer questions now or by email if somebody wants to contact me. Back to you, Susan.
Thank you so much, Steve. That was great. And let me open the floor. If you want to stop sharing your screen, then everyone could kind of see each other and see what questions people might have for you. Feel free to just unmute and ask a question.
While people are thinking of questions, I'll tell an anecdote that I think is humorous, but and ironic at the same time. So, as I mentioned, my early work was helped along by some really great people in Asia, and colleagues of mine from in Hong Kong, from Chinese University of Hong Kong, invited me to give a presentation about this work to them in Hong Kong. And so I combined it on another trip, and I gave the talk, much like I just did. And my host, who's a well-known botanical expert, a surgeon by training, turned to his colleagues and said, you know, we really should be embarrassed, if not, you know, this is a wake-up call, he said. Here it takes, you know, an American to come talk to us about plants that we have a tradition of use for, you know, that grow in our forests. And, you know, the reason that he had to say that was because, unfortunately, there was a preponderance, if not exclusive, training of Asian, in particular, I'm talking particularly China, Hong Kong, Korea, Taiwan. It was for people interested in going to medicine or into neuroscience or to any medical field for them. But there was a preponderance of emphasis on training in Western medical schools, Western laboratories, you know, and it was only more recently, in the last 10, maybe 15 years, that young trainees are getting experience in the world of botanical medicine and learning techniques by which to study them, like I did. I mean, in other words, using modern methods to study botanicals. So it's been an interesting, you know, kind of journey to see their transformation in the way they approach botanicals and in what happened here. As I mentioned, when we started this at Harvard, I mean, I can't tell you how many bewildered colleagues I had, and wondering, like, had I gone nuts? What was I doing? Um, uh, but again, coming back to the unmet need, I knew my patients and other patients were suffering, and I don't care if it comes from a plant or from a chemistry lab. If it helps somebody stay alive and thrive and be able to live a productive life, that's what counts.
I have a question. Is it okay to just interject, Susan?
Sure.
Okay.
Please, please.
So you're like a drug detective, and I wonder how often is it that there is a cultural phenomenon or a cultural use that precedes the identification of a forest botanical, or how often is it a knowledge of the plans that directs you to identify a potential use?
Yeah, it does. I've come to see it does go both ways. But what made it possible in my work is that it was the effectiveness of a plant for epilepsy doesn't require sophisticated diagnostic testing. It should be visually obvious in terms of reducing convulsions. Now, what they didn't know back then was that there are other types of seizures than just convulsions. But I mean, there's a story of a convulsion in the New Testament. I mean, the history of epilepsy goes back to the beginning of recorded history. The Greeks wrote about this, et cetera. So, but as opposed to, say, a plant for gastric cancer, how would you, there could be a swelling, and it's not as obvious for some other medical therapies or medical uses that a plant could be effective. So for those hidden medical conditions, it's more often the case that you're looking for a particular mechanism of action. And if you're looking for that in a plant, then you might stumble across that plant. And sometimes it goes both ways simultaneously. I tell the story about the treatment of malaria. So there's a plant, I forget the Latin name for it, but a compound called artemisinin is isolated from it. During the Vietnam War, the leader of North Vietnam contacted Ho Chi Minh, the leader of China, to ask, is there anything in the traditional Chinese medicine armamentarium for malaria, the treatment of malaria? And sure enough, there was this plant that was used for a condition that sounded like malaria, in ancient textbooks. It was then brought into North Vietnam, and the compound in it is effective for malaria. It was then later grown in East Africa, near where malaria epicenter is, as an example of a microeconomy that can develop from this work. So that was a cultural tradition meeting an ongoing need, even before it was clear how artemisinin worked. So it seems like each one of these is its own unique and interesting story. What I've tried to do is set up a standard process, and provide an example for how one could think about this work. I think the mistake that's made, though, that I see is people go straight from the tradition of use into clinical trials, and those invariably fail. And it's not because the treatments necessarily fail, it's that they pick the wrong dose, they pick the wrong disease, they pick the wrong type of patient, et cetera. It's problematic to do it without an intervening animal study step.
Thank you. Other questions? Oh, sorry, I was muted. Feel free to just unmute yourself if you want to. I wanted to just also highlight the interesting kind of puzzles that you have to figure out as a physician or as a scientist. And thank you for sharing the details about all of that. I have two small questions. Have you thought about compounding any of these botanicals together since you found so many compounds with efficacy? And my second question, which I think relates to your talking about how in Asia, they're just starting to integrate traditional training into medical schools. I think it's interesting here, we probably have a lot of analogs of some of these Chinese medicines, but we're always going to China looking for these traditional therapies. And maybe there might be some opportunities to look at what are some of the botanicals that are growing here that might have potential.
Exactly. So thank you. For the first question, yes. Again, coming back to the principles of traditional Chinese medicine, and Ayurvedic for that matter too, it's hardly ever one single herb or one single formulation plant that is used as a recommended therapy. More often, it's what are called formulas. It's just, if you go to a traditional Chinese medicine doctor and they write you a prescription, it's called a formula. You take that to the traditional Chinese medicine pharmacist, and what they do is pull different things from different boxes according to the prescription, and the prescription also indicates the weight ratios of all these. So, 2 ounces of this, 4 ounces of that, 6 ounces of that, and the principle in TCM is that some of these are meant to treat the symptoms, some of these are meant to support the health of the individual, and some are meant to mute the toxicity or side effects or potential bad effects of both of those other groups. And it gets pretty fanciful, and TCM is what these different categories are called, but by contrast, Western medicine, we often focus exclusively on what we call monotherapy, or single compound, single molecule therapy. So we've actually conducted some experiments looking at adding, we studied one particular herb. It's actually whatchamacallit? Oh God. I'm trying to think of the name of this plant. I can't even think. Oh my goodness gracious. Oh well. Anyways, we studied one particular plant that from which there are several compounds that would appear to have a benefit mechanistically in epilepsy. Orchid, that's what it is. It's a type of orchid. Sorry. I think I need some huperzine. And we created some extracts from this orchid plant in a laboratory in Singapore at a university called Nanyang Technological University. And they're interested in this orchid because it's a major export crop for Singapore. So however you can keep it fresh and in the export process, that's of major value to Singapore. So we were able to isolate individual compounds from this orchid plant, and then tested the compounds individually in the animal models. And then we tested combinations. And at first we didn't quite know what the ratio should be of the different amounts in the. But no matter what combination we what weight proportions we use in terms of combinations, the benefit was about the same, but the toxicity was less. So we found that. So we've only begun to dabble in that aspect of it, but I think it gets complicated pretty quickly to try to figure out what combinations should be used and at what proportions they should be used. As far as your second question goes, we've had a reliance in our work on particularly in traditional therapies in China, Japan, and India, because those are the most well documented over the centuries. But when you start to get into it, one of the fun things you find out is that every culture, every group of people, as you can define it, has an herbal tradition for treating various things. And whenever I used to travel quite a bit to different countries, I would look up, what had been written about traditionally coming out of their own territory. And even Native Americans had a tradition of use for epilepsy. And I talked to some herbalists in Native American groups and found out what they were using and how they prepared it and so on and so forth. We've focused so much on huperzine lately, we haven't had a chance to go back and study those rigorously. But what we need is some way of gathering all this historical and experiential data together into a single compendium. We have started a website called Naturopedia, that's meant to be that kind of repository. And sometimes the answer isn't in plants that you would take independent of your diet. Sometimes the answer is in the plant that you eat in your diet. And unless there's a question, I can give a quick story about that. So a neurologist from Kuwait contacted me, and he said there's something very interesting that children who live near the Nile River in Egypt have a lower prevalence of epilepsy than children who live in Egypt away from the Nile River. And he spent a fair amount of time trying to figure out why that was. And he eventually, talk about detective work, he eventually figured out that the diet of children who live near the Nile River was much more, had much more of broad beans in it, a type of bean, broad bean, for the reason that it grew there. And it was digested, it was eaten every day. It was part of their lunch meal and so forth. So the diet was rich in broad beans. I thought, well, there's the answer in broad beans. So we collaborated on testing broad beans in animal models of epilepsy and trying to understand, sure enough it worked, and then trying to figure out how it worked. And it turns out that it inhibits another excitatory neurotransmitter called glycine. So, yeah, sometimes it's in the diet. And of course, Susan's very interested in the diet for epilepsy because of ketogenic diet, which isn't so much a matter of what food is in the diet, but how the diet affects body and brain chemistry, we think, and how that lessens the likelihood of seizures. So, yeah, go ahead. I'm sorry.
I was just going to say, yeah, great. Thank you. And, there may even be compounds in plants in this area that are the same compounds that are found in traditional Chinese medicine. We just haven't really isolated all that yet. Related to that, there's a question in the chat. Could you expand more on Native American contributions to medical treatment? Has anything been learned about tribal nations here in the Northeast, and where could we learn about this?
Oh, right, great question. No, I don't consider myself at all an expert in this, but if you go to the website for the Tucson Medical School, and I forget the exact name of the Tucson Medical School, there is a integrative medicine division there. In fact, I think that's where Andrew Weil was from, I believe. And there's another faculty member there who is a Native American herself and has studied this. And I would reach out to her. If I assume she's still on faculty there. I'm blacking on her name. Somebody might want to look it up while we're still talking. There are a couple other neurologists I know with an interest in this. But I, would not, I would defer you to others who have looked at this.
Mm-hmm.
They, you have to have a long history in a region with a very, stable supply of particular plant-based products in order to develop that kind of tradition of use. And so, if any group, holds the key, the secret to understanding potential medical uses for plants that grow native in the Americas, it would be those who have, who've lived here for generations that have been able to accumulate that experience.
Yeah. Thank you.
I should also mention that when it comes to natural products, we could also think about things in the sea, marine-based. And that opens up literally and figuratively a whole new world, a whole different world of potential therapies as well. And what's interesting is what may be viewed as poisonous can also be potentially therapeutic. So for example, I showed a picture of the foxglove plant before. It's poisonous. You don't want to be munching on that willy-nilly because it can paralyze the heart. But it's the same action on the heart that gives rise to its medical use, and its action in the brain at those same kind of doses can be as an anti-seizure medication. Likewise, from the sea, you have poisonous peptides that come from conus snails. And these peptides is what the snail uses to paralyze fish. But the reason that they paralyze fish is because they have an effect on the nervous system, and those effects on the nervous system can be potentially therapeutic and have been used. In fact, there's a therapy out now for spasticity and multiple sclerosis that's based on one of these peptides. So while there's no literal forest that grows in the sea, there are untapped resources there as well that could be, brought to bear as therapies. And Susan's mission to save forests, which is multi-pronged, but one of which is because they may, be the key to healing ourselves. The same, you could think of the same strategy when it comes to saving the seas from, wholesale changes in sea, pH and temperature and so on and so forth that could jeopardize, the therapies that have not yet been discovered.
Yeah. Thank you, Steve. Other questions?
I was just going to mention, so my family's originally from Haiti, and there's this sort of, there are some ideas about zombification, and there's some evidence that ingredients from pufferfish, particular tetrodotoxin tetrodotoxin
Yeah.
that causes paralysis has been used in some rituals that give the appearance of zombies in the subjects who take it. But yes, the C.
Yeah. And I mean, neuroscientists use that toxin all the time, in their work. And there are other, these are very small peptides that have an unbelievably potent effect on nerve function, or nerve receptor function. uh, so, I think what we're trying to say, particularly, for students, is that there's a whole world out there to be discovered. And maybe I'm an example of somebody who, but I think it's a pretty typical example that you think you're starting off on a career path and you have it all laid out and it looks like it's going to be a straight line to the finish line. And at some point you take a detour, or, if you like to sail, you tack this way, you tack that way, and my advice to you is to expect that to happen during your career, and to pause and think to yourself, is this an opportunity to pivot and transition to something that could be really fun, exciting, and productive? Because these will come along, but if you don't recognize it, you'll ignore it. And that's the way I think. I mean, when you learn science, it doesn't come across that way. It seems like it's a progressive, unidimensional advance in knowledge and application. But when you talk to the people behind those advances, this is a typical story where, they zigzagged, and some of it, if not all of it, was because they came up with an unexpected result, or they came up to something didn't make sense, or there was something unexpected, or maybe they initially ignored that, but when they thought about it, that held the key. And there's many, many stories of that accidental discovery occurring. It's not all by design. But what you can design is how you take advantage of those opportunities. So I'm just trying to convey a little advice to you.
Thank you. Yeah, I was also going to say, well, that's also why, I'm a real fan of the liberal arts tradition, because it kind of helps you to think in a number of different ways that can, I think, give you the flexibility to make those connections and be able to think about pivoting in different ways and connect with different types of people. And I also like how you shared that the teamwork that was involved in kind of pulling all of this together. If you didn't have the open-mindedness or whatever to bring all those pieces together, you would never have been able to see this to fruition. Any other questions for Dr. Schachter before we thank him for his time? Let him get on with his afternoon. All right, well, Steve, this was really, really special treat for us today. And I will be in touch. And I want to thank you. Thank you again. Give you a little round of applause.
Appreciate that. Thank you. And good luck to everybody.
All right. Thank you, Steve. And thank you, everyone, for joining.